Hello, I'm Noah Holkenborg. I'm a second-year student here at Grand Valley Indiana's College. I'm double majoring in cell and molecular biology and biology with an emphasis in ecology and evolutionary biology. Today I'm going to walk you through some of the research that I've been working on for over a year now. In that time, I've presented it at multiple conferences, including GVSU's own Student Scholars Day, the Michigan Academy of Science, Arts, and Letters, and the West Michigan Regional Undergraduate Science Research Conference. What I'm going to dive into today is a poster that I prepared for some of those conferences, and after that I'm going to touch on some of the research that I have done since then and the future directions that this project might go in. The research that I'm involved in focuses on the branching patterns of trees and how we measure those branching patterns using photographs. I'm doing this research with Professor Gary Greer of the biology department here at Grand Valley. The poster that I'm presenting today specifically focuses in on using this new methodology to look at the optimization of ginkgo trees in comparison to some other native species. Before we dive into that, a little bit of background. At the basic level, to survive, trees need to do certain things. They need to transport water and nutrients, they need to grow, they need to get sunlight, they need to accomplish certain tasks. Now, they can be optimized for these tasks. And the more optimized a tree or species in aggregate is for a certain task, the better it does it. But that often comes with risks or trade-offs. For instance, if a tree is trying to optimize for having really big branches, that might benefit it, but it might mean that there's a higher risk that those branches would break off because they're larger and heavier. Now, to determine how well a species is optimized for a certain task, we put data about it into a corresponding mathematical model that will tell us how well it is optimized for that specific task. Now, in our research, we focused on three models. We looked at Murray's Law, which looks at hydraulic efficiency, and it's the simplest model we looked at. It is based off of area being conserved at branching points. Basically, below a branching point, you would take the area of that, and if perfectly optimized, that area should be equal to the areas of the two branches right after the branching point put together. The next model we look at is the uniform stress model, which is about the structural support of the limbs. Now, if a tree were to over-optimize for this model, it would put its branches at risk of breaking. So, in this model and the others, a safety margin is often kept. That means that a tree could be perfectly optimized for something, but it would be intentionally under-optimized to keep it safe, because the closer you get to that optimization, the more risk it exposes itself to. The final model that we look at is the West-Brown-Enquist model, which predicts for the total size of the tree, how massive it is. And if a tree were to over-optimize for this, it would be more susceptible to wind knocking it over or breaking parts of it off, because it is simply more massive. Now, these models are nothing new. They have been studied in the past, but that study has primarily been with destructive methods, cutting the whole tree down and measuring at the branching points, or cutting branches off of multiple trees and making measurements that way. And while those measurements are very accurate and you get good data from it, that is costly and takes a lot of time and energy. And it also limits what type of questions that you can ask. For instance, you can't see how trees change over time if to get data from them, you have to cut them down. It also means that to do any sort of large-scale study could be prohibitively challenging. Now, the methodology that we are trying to develop involves photographs, simply getting measurements by taking pictures of trees. Now, this will probably lead to data that is not as accurate as cutting down the tree, but it means that we could gather data from many more trees in a shorter amount of time, and we could ask somewhat different questions. For this study specifically, our field methodology was fairly simple. We would find a species we would find a tree of the species we were interested in, take a couple physical measurements like dBH, and then we would take some scaled photographs, one of the entire tree and one of the first node or first branching point. We would then take these photographs, which had a scale bar in them, and put them into a program called ImageJ, which we can use to make some measurements. We measured the mother diameter and the two daughter diameters before and after the branching point. And we also measured the angle in between those branches. Then, using that data in each model, we ran a regression compared to the observed data for the mother and daughter attributes. And the predicted values per these different models were generated using residuals from the observed data regression, so that our predicted data wasn't as perfect as would be predicted. If we left it as is, it would be very, very easy to generate statistically significant results, simply because nature is messy, and so if you expected it to follow a perfect line, you would never get that. So then we took those results and we ran an ANCOVA comparing the slope of the y-intercept of the observed regression and to our predicted. Now, what we found was that we could see differences in optimization, which is a really good sign. Our photographic data allowed us to see where Ginkgo deviated from the model's predictions. Specifically, the largest deviation was in the West-Brown-Enquist model. You can see that for the West-Brown-Enquist model, the slopes of the observed and predicted differed significantly. And this means that Ginkgo differed from the model's predictions most in terms of optimization for total mass, because the West-Brown-Enquist model is about total mass. Ginkgo best fit Murray's law and the USM. So that means that Ginkgo seems to be sacrificing its total mass for hydraulic efficiency and some structural support of the branches. Now, we don't know why Ginkgo keeps such a large safety margin for its total mass. We don't know why it's so small. But one of our hypotheses is that our Ginkgo were mostly found in cities, in fact, almost exclusively found in cities, because Ginkgo isn't native to Michigan. It's only really found where it has been planted. And so it doesn't have to compete with nearby trees. There aren't neighbors that it has to outgrow to get to the sunlight. So there isn't as much kind of competitive pressure for it to be tall. But being in the city also means that it could be exposed to more wind. There aren't as many neighbors there to shelter it from the forces of wind, which also could be a good reason not to be as massive. We can also see how Ginkgo compares to some other species that we looked at. And there are some interesting differences here. We can see how it compares to sugar maple and American beech. If we look at sugar maple, it seems to be slightly under-optimized, but all of the three models we looked at seem to be about the same. Now, my kind of interpretation of this is that sugar maple is trying to, be successful in all areas. It's not perfectly optimized for everything, but it's not severely lacking in any areas. Then, in contrast, if we look at American beech, for Murray and USM, it is very under-optimized, but for the West-Brown-Enquist model, it is very close to optimization. And this seems to indicate that perhaps in the beech that we looked at, hydraulic efficiency and the support of the branches are less important than, just being massive, than total size. And then again, we can see the optimization of Ginkgo. We see that for Murray and USM, it's fairly close to optimization, but it is lacking in West-Brown-Enquist. It's smaller, it's not as massive. So looking at all these together, we can fairly confidently say that the photographic data that we gathered in the scaled photographs gave us usable data. We could see differences within a species and how it differed for different models, but we could also see differences between species. And these results are what we would expect. Slight under-optimization, keeping a safety margin, is consistent with what is already known about tree biology. And so, as a proof of concept, which in some ways is the most important element of this study, it seems that photographic data is usable. So, in that ways, we are fairly happy with how our experiment went. We can see differences in optimization just from taking pictures of trees. We don't need to cut them down. And so what we gathered about Ginkgo is that it seems to minimize its risk of buckling in the wind by not being as large as it could be, while excelling in other areas like hydraulic efficiency. Now, this specific study is mostly working on the model. What we found out about ginkgo and the other trees is interesting, but the most important thing is that we seem to have usable data from photography, which is new to the field. All this research before has been done destructively, so if we can come up with a way to do it by simply taking photographs of trees, that would be really interesting and allow us to do a lot more work. Now, the research that I just presented is actually somewhat old. This is what we worked on last year, and since then we have done a whole lot of work. I have continued to work on this project, and over the summer I was actually a paid remote research assistant. So we decided to develop a plot in Grand Valley's ravines. Basically, instead of specifically measuring ginkgo, I went out and measured every tree in a plot. I measured 200 trees this summer and took photographs of all of them, scaled photographs. And we have yet to analyze that data. That's something we're in the middle of right now. But the reason we are doing that, the reason why we have established a plot is that we hope to try and tease apart how much of optimization is dependent on the species of the tree, like I talked about in this poster, or how much of it is about where the species is located. We hope to establish another plot in another location at some point to compare. Instead of just running our models with one species, maybe we would run them with all of the trees in one area to see if there is some general trend of optimization in an area regardless of which species are there. This plot also has many other avenues we could go through to try and understand different things about the branching patterns of trees. Part of the reason we're establishing it now is maybe in 10 years someone will go through and measure all these trees again, and we'll see if their optimization has changed over time. Another area that we're working on is destructive confirmation of this photographic data. What we found so far is great, but it is not concrete as establishing it as a valid methodology. What we really need, our next steps, are to take photos of a tree, take our measurements from that, and then cut the tree down and take the measurements the old way and compare. If there is a drastic difference between those two, well, it might mean that our photographs don't actually produce as good a data as we had hoped. But if we can get similar results, that means that we don't have to cut down trees to get usable data. Those are some of the things we're working on currently, and in the future I'm planning to stick with this project for the foreseeable future. And it's really exciting. I really enjoy it. So I hope that you enjoyed this little presentation, whether you are watching it for the biology or you're here from the Honors College to hear about some of the research happening around on campus. If you have any questions or you're interested in this project, feel free to reach out to me. I would love to talk more about trees or just Grand Valley and the research going on here. I'll put my contact info up so that you can reach me, but I hope that you have a wonderful rest of your day, and thank you for watching.